And and so that's been our game plan. It's also an opportunity for us perhaps to interact with members of the community. And so we, as time goes forward, we expect to see more and more members of the community taking advantage of these. Our first luminary speaker in 2012 was Ben Carson. He was followed by David Cooper, and then Ken Ledberer, and finally Ron Harden. And so I'm going to turn the podium over to Dr. Rick Hodinka, who's going to introduce today's speaker.
So, good afternoon, everyone. It's truly a pleasure, a great pleasure and honor for me to introduce Dr. Doug Wallace. We overlapped at Children's Hospital of Philadelphia and the University of Penn for about 4 years. You came around 2011 before I came here. So, and if you don't know it already, Dr. Wallace is truly, truly a pioneer in the study of mitochondrial DNA. And more than 35 years ago, he and his colleagues founded the field of human mitochondrial genetics. And by way of introduction, let's look a little bit about his education. So in 1968, he received his BS in genetics and developmental biology from Cornell. He then went on to Yale in 1972 to receive a master's in philosophy and microbiology and human genetics. How much micro did you use? A little bit. And in 1975, he got a PhD at Yale in the same department. He then moved on, or stayed at Yale actually, in the Department of Human Genetics to do an NIH postdoctoral fellowship, and then did
Bacteria has a membrane potential, a capacitor of 0.2 volts. So 10 to the 17th times 0.2 volts is the equivalent of a lightning bolt. That is the amount of energy right now that you're using to sit here and listen to the lecture. The difference between being alive and dead is when this membrane potential collapses. Or as the Greeks say, when you stop breathing. So if you don't think that's real, just stop breathing for the rest of the lecture. Okay. So all of this process is to maintain potential energy, because potential energy allows you to do everything that you want to do. Now, you could want to do some chemical work. Sometimes potential energy is not so good for that. So you can use this enzyme, the ATP synthase, or complex 5. Protons flow through the ATP synthase. The energy that's released is used to condense ADP and phosphate to make ATP. They then are exchanged by the adenine nucleotide translocator out through the voltage-dependent anion channel, and there the ATP can be used to do work. So now you've coupled phosphorylation with oxidation, hence oxidative phosphorylation. Now, it turns out everybody in this room has a little bit of different efficiency of pumping protons out or converting them to ATP. We call that the coupling efficiency. And as you'll see, that's very, very important in understanding how. Okay, so if you inhibit the electron transport chain, then electrons will build up here. And they are electronegative enough that they can be put right on O2 to give you an unpaired electron, superoxide anion, first of the reactive oxygen species. These are very toxic because they want another electron on that outer orbital, and it'll take it from lipids, proteins, or DNA. And that is then an oxidation process. So we have an enzyme, manganese SOD, that will take two of these and condense them into hydrogen peroxide, which is more stable. But if it runs into a reduced transition metal, it can get another electron, and that gives you a hydroxyl radical with an unpaired electron, and that's even more potent oxidizing agent. So the reactive oxygen species that you read about and take vitamin E, vitamin C, and beta-carotene for are all primarily coming from the mitochondria. Okay? Mitochondria also is a major sink for calcium, and calcium is the major inorganic regulator of a large range of biochemical processes. And finally, the mitochondria has a self-destruct system called the permeability transition pore. And it involves an outer membrane protein, which we think is the TSPO, the intermembrane channel, dental channel, which Paolo Bernardi thinks is the ATP synthase. It's regulated by the adenine nucleotide translocator. This system is normally a closed door, but when the membrane potential gets low, high energy phosphates decline, oxidative stress gets high, or calcium load becomes excessive, they impinge on this, and it goes from a closed door to an open channel. When it does that, it shorts the membrane potential. Fluids flow into the mitochondrial matrix, which is they release all these stored protein genes. And what are those? Small and large ribosomal RNA, 22 transfer RNAs that punctuate all of the different larger coding genes, and the 13 proteins. ND1, 2, 3, 4, L4, 5, and 6 for complex 1, cytochrome B for complex 3, CO1, 2, and 3 for complex 4, and ATPase 6 and 8 for complex 5. And then there's this control region that regulates replication and transcription. So this is the wiring diagram of the power plant, and it's inherited from one and only one parent, so that the only way it can change is by sequential mutations along radiating maternal lineages. Okay? And each new mutation then is tested against the existing variation. There are now 3 classes of clinically relevant mutations: recent deleterious mutations, ancient adaptive mutations, and somatic mutations. So what are some of the recent deleterious mutations? These are ones that would arise in the maternal lineage and give maternally inherited diseases. So this is a mutation in the tRNA leucine gene at 3243, and if you have about 10 to 30% mutant mitochondrial DNA, you can either get autism or type 1 or type 2 diabetes. If you have 70% mutant tRNA, you will get neuromuscular disease, and if you have 100% mutant mitochondrial DNA, you will die of Lee syndrome in the neonatal intensive care unit. Mutation in the tRNA glutamine gene predisposes you to late onset Alzheimer's and Parkinson's disease. Mutations then in the tRNA lysine gene will give you kind of epilepsy. There are literally hundreds of these protein synthesis mutations. You can also have mutations in polypeptides, mutation in the MD4 gene at 11778. That's, you're fine when you're young. Suddenly, in midlife, you'll lose vision in one eye, and then in the other, Leber's hereditary optic neuropathy. Mutation in the MD4 gene, 14484, gives you the same phenotype. At 14459, which is more severe, if you're heteroplasmic, you get optic neuropathy, but if you're homoplasmic, pure mutant, you get generalized dystonia as a child. Mutation in the tRNA ATPase 16 at 8993, at 70% mutant will give you retinitis pigmentosa. 80% mutant gives you oligopontocerebellar atrophy. And 100% mutant kills you as a baby, as Lee syndrome. Okay? So we have a quantitative genetics with differential clinical phenotypes. So there are literally now almost 500 pathogenic mitochondrial point mutations. You can find them at our website, Mitomap. The other class of mutations are these ancient adaptive variants. That variant in ND1 is found in 3/4 of sub-Saharan Africans. Variant H is found in half of Europeans. Variant A, B, C, and D arose in Central Asia. They crossed the Bering Land Bridge, colonized the Americas. These allowed changes in mitochondrial energy metabolism, allowed our ancestors to adapt to different places in the globe. And then we accumulate somatic mutations, as we talked about here. Okay. So, once you begin to think energetically, then you can say, oh my goodness, then all of anatomy is really about energy. So there are organs that have high energy demand, like the brain has 2% of the body weight, but uses 20% of the oxygen. Therefore, a 5% change in mitochondrial energy is going to give you a massive headache. Also, the heart, muscle, renal, and endocrine systems are affected because they're high energy demand. There are energy storage tissues. White adipose tissue stores fat to make ATP. Brown adipose tissue stores fat to make heat. There's an energy homeostasis tissue, the liver. Its job is to regulate glucose. Did anybody ever ask you why? Why do we care about regulating glucose? Because that's our connection with sunlight. We have to have our glucose level at a certain level. And then we have the pancreatic beta cells and alpha cells that monitor that glucose level. And when we have high glucose, the beta cells send out insulin and say shift metabolism toward glycolysis. Or when we are starving, the alpha cells sense low glucose, send out glucagon. What does it do? It induces fatty acid mobilization and upregulates mitochondrial oxidative phosphorylation to burn the energy so we get through a fast. So our anatomy is based on energy. Okay. So this idea then that mitochondria might be very important in common diseases leads to the question of how could a degenerative disease actually occur? Well, we're born with a whole bunch of cells in each organ. And the mitochondria, as it's respiring, is going to make ROS. And the ROS, over time, is going to damage the mitochondrial DNA because it's mutagen. And eventually, the mitochondrial DNA mutants are going to be amplified, and that's going to be bad enough to cause an energetic defect, to trip the permeability transition pore, and cause the cell to die. Okay? Now, this is an ongoing process going on in your body all the time. So, why don't you just die outright? Because you're born with extra cells. So, this process is going on, and as you age, you begin to decrease the number of cells. That's fine because you have an excess of cells until after post-reproductive age, which is about 50, and then you begin to lose more cells than you can afford. And when you lose cellularity, then you begin to get long-term post-mitotic cell damage. So here is a family that we studied years ago while we were at Emory. This mother brought these two young people in. She was very concerned about this little boy because he was in her arms and he was hypotonic and not responding, had had what seemed like a mild fever, and she was followed by this little boy that was going around the hall like this, and cerebellar ataxia. To see it coming in. Okay. So that's Lee syndrome, that's cerebellar ataxia. Why was she concerned? Because this little boy from a different husband died of exactly that, and suddenly she saw the symptoms, so she came running to us. Okay. We took a history. We looked at her eyes. Nancy Newman, my colleague, looked at her eyes, and she had salt and pepper retinitis pigmentosa. We looked at her mother, so did she. She had had two different husbands, and these two girls also had salt and pepper retinitis pigmentosa. History showed, we could never see this person, but this person was in a nursing care. He was mostly immobile, severe mental retardation, and as you'll see, he had oligopontocerebellar atrophy. To make a long story short, there was a mutation in ATP6 gene, a T to C gene transition at 8993, changed leucine at codon 156 to arginine. Okay, so what did this man who had the most dramatic phenotype look like? So this is his retina, and you can see spicular RP. Okay, and he's, oops, the macula is being lost. This is an MRI of his head, and you can see that the cerebellum has shrunk to about a third of its size, and the brainstem is about half. Oligopontocerebellar atrophy. Okay, so that's why he was sick. Why, biochemically, was he sick? So we took the cells from the mother, because she turned out to be 70% heteroplasmic for that mutation. We enucleated the cell with a density gradient, took the cytoplasmic fragments with her mitochondria, took another cell of a different nucleus, cured it of its resident mitochondrial DNAs, fused this cell to that, so we now have this nucleus, but with these mitochondria. Okay? Then we can then take these cells and segregate out the mutant and normal mitochondrial DNA. So we have two pure forms. So then we can take these cells and put them in a closed chamber and measure the oxygen as the oxygen is used up using a substrate, respiration. So this is the normal wild type hybrid. We've added the mitochondria, and we're just looking at the use of the oxygen. So we've added the substrate, which is complex 1 subunit, and you can see that the respiration goes on. Now we add ADP, and it's using that membrane potential. So the electron flow has to increase to keep the membrane potential up, so the oxygen consumption goes up. Then when all the ADP is phosphorylated, it goes back to a slower level, add ADP goes up, goes back to slower level. Now we just punch a hole in the inner membrane, and all short circuits the membrane potential, and the electron flux is maximized, still trying to pump out the protons. Okay, that's normal. So what happens when we have pure mutant? Well, you can see that when we add the ADP, yes, it increases the rate, but it's much slower than this rate. Okay? And in fact, it's about 70 to 80% slower. So therefore, a 70 to 80% reduction in the rate of ATP synthesis was enough to kill that baby with Lee syndrome. Okay. All right. And this enzyme is quite amazing. This is the ATP synthase. And it's a wheel that exists inside the mitochondrial membrane. This is the inner membrane. And it has an axle that spins inside this barrel of three copies of each of 2 proteins, alpha, beta, alpha, beta, alpha, beta. And they're bound to this structure and immobilized. So the barrel is immobile. The wheel spins around, spins the axle, and the axle is cooked. So as it hits the beta subunits, it changes their conformation to bind ADP and phosphate, to push them together, and then to release them. 3 steps. Every time the wheel spins, makes an ATP. Okay? So the protons are flowing through this system and driving the spin of the wheel that makes the ATP. Okay? So how did the mutation cause the problem? This is that static system called the stator. That's the part that, this is this part and holds this. That's the stator. That's the mitochondrial DNA encoded in subunit 6. And this is one of the subunits of the wheel, one of these. Okay? And these have a specific carboxyl group that binds the phosphate, spins all the way around, and releases it. So what happened in the mutation, a leucine, which means that there's a hydrophobic region that allows the proton to bind to the carboxyl group, is mutated to a positively charged arginine. And that arginine then makes a salt bridge with the glutamate and locks the wheel. So the wheel stops, no ATP synthesis. Okay? Very simple idea, but devastating pathological. Okay? So it's an example of a maternally inherited heteroplasmic mitochondrial mutation giving highly variable phenotype. This is another disease. This is Leber's hereditary optic neuropathy. Mutation in the ND4 gene, hormone 778, gene A, arginine, codon 340 histidine. And this person went blind related through this woman, to this woman that went blind, through this woman, to all these men that went blind, to these men that went blind, through this woman, this man and woman that went blind. Okay, so there's maternal transmission of blindness, but everybody didn't go blind. Why is that? Well, first of all, you can see that there's a predominance of males going blind relative to females, 4 to 1. In fact, that's exactly the ratio we see in autism, which is why I believe autism is a mitochondrial disease, and we're actively studying that. But anyway, the point is that there is variable expressivity with a strong sex bias, and that is very characteristic of mild mitochondrial disease. But why is there variability? And it turns out what causes variability is the context in which the mutation occurs. So these are 3 common mitochondrial DNA mutations that give blindness. This is the more severe, 3460 and ND1. 11778 is milder, and ND4. 14484 and ND6 is the mildest. And they represent a reasonable portion of the European patients. So if this is milder than this, why do they get the same blindness? Because this can cause blindness when it also interacts with the right kind of mitochondrial DNA. So this is a lineage we call J, and I'll come to that. So the more severe mutation, J is 10% of the European population. So it doesn't matter whether 3460 occurs on J or not, you'll still go blind. But if you have the milder mutation, then about 1/3 of all the cases are also J. And if you have this very mild mutation, in fact, 3/4 are J. So therefore, the milder mutation, the more important it is that the right background occur. That is, there's an interaction. Here's another mutation. This is an ND1 mutation, 3394C, and it can give you the same expression enhancement for these mutations. That mutation, ND1 T3394C, changes a tyrosine in codon 30 to histidine in the ND1 gene. So where does all this come from? It comes from this. And that is, the mitochondrial DNA is transmitted from mother to daughter. And the only way it can change is by sequential mutation. So if I sequence all the mitochondrial DNAs of each of you in the room, the number of nucleotide position differences between each of you is proportional to the time you shared a common mother. So all we did was sequence the mitochondrial DNA from Aboriginal populations around the world, and knowing their location and their sequence differential in mitochondrial DNA, we could reconstruct migration women, as mentioned. And the answer to that is mitochondrial DNA traces back to a foundation in Africa about 150,000 years now, but it was originally 200,000. And that's what we find in the Khoisan people of Kalahari, so-called Bushmen. And then L1 and L2 are two different pygmy lineages. They look exactly the same, but they arose 50,000 years different in time. Then there's L3, which is the sub-Saharan Africans. And then in this area in Ethiopia, two lineages arose, M and N. I'm sorry, that's not very clear with this light. And these are the only two mitochondrial DNAs that left Africa. So there are hundreds of African mitochondrial DNAs, only two left Africa. M stayed in the tropics and were all the way down to Australia. But N left and went into the temperate zone. Gave rise to I, W, and X. These are just letters we gave them as we found these lineages. Or through R, H, J, T, U, U, K, and V. Then later, some of M gave rise to European lineages, Asian lineages, C, D, G, and a whole bunch of M's, as you'll see. And lineage A arose in the N lineage. And A, C, D, and A became enriched here in Chukotka, and they were available 20,000 years ago to cross the Bering Land Bridge to colonize the Americas. Then later, M moved along the coast to colonize central, southern North America, South America, and Central America, and M also colonized all the Pacific Islands, and then X somehow, at 15,000 years ago, got to the Great Lakes. I think they invented the airplane. I don't know. Anyway, that's still a mystery. But the point of this is that there is a high geographic correlation between mitochondrial genotype and position of where the people, ancestors, were. Why is that surprising? Because nuclear genetic variation is panmictic. It changes in frequency, but all the same major alleles are found in all populations. Not true in the mitochondria. So why would that be? Because here in Africa, if you're a woman and you want to reproduce, you need to run away from lions. Running away from lions means that you need to have the maximum amount of ATP per calorie burned. A calorie is a unit of heat, right? So you're going to burn the ATP, you're going to burn the calorie to make ATP. That means you have to be tightly coupled. That is, you have the maximum protein output and conversion into ATP, minimum heat. So that's best in the tropics. But up here, the lions froze to death. That wasn't a problem. The problem is you're going to freeze to death. How do you avoid freezing to death? You have mutations in your mitochondrial DNA that decrease the efficiency. Now you eat more calories for the same amount of ATP, but a calorie is a unit of heat. More calories you burn, the more internal heat. regenerate. And so, in fact, uncoupling is the best way to deal with chronic cold. And so we believe that a lot of these mutations changed our physiology of our ancestors and allowed them then to adapt to a whole bunch of different environments. They also changed ROS, which is very important in I can't remember exactly how to say the other one, but this is a group of humans that is found in the Altai of Siberia, represented by one tooth and one finger bone. But those whole genomes have been sequenced. So there is 3 to 5% Neanderthal mitochondrial DNA, nuclear DNA on average in everybody in this room. Yeah. If there were people here from the South Pacific, they would have up to 7% of the, I think it's Dosninian individuals. Okay, so there are 3 independent bipedal Homo populations. They did interbreed. That is credit to the people in Leipzig for proving that, which is amazing. The question is, was there mixing of mitochondrial DNA? Or was there mixing of the Y chromosome DNA? No. We proved that years ago. So each of these species, their mitochondrial DNA is totally distinct and non-overlapping in their variation. So this was another topic I was going to talk about. We think that mitochondrial DNA variation allows regionalization of populations, and that it is the driving force for speciation. And so then that would predict that if there's a different population, they would all have a different convergent mitochondrial DNA, and that's what we see.
Dennis?
I had a question of DNA or mitochondrial DNA replication. If you have heteroplasmy, do you see competitive advantage in some of these mutations such that the disease may be variable?
Yes. So the question is, let's say that, if I may rephrase, we start out with a heteroplasmic zygote. Let's say it was 25% mutant, 75% wild type. Could there be a time when it would go to more mutant? And the answer is emphatically yes for certain mitochondrial DNA mutations. So for mitochondrial DNA deletions, which I didn't talk about, these occur probably in the egg, and a few molecules get deleted. And then those molecules get dispersed as the development occurs. So you get, every cell gets some deleted. What you find is that in post-mitotic tissues, over time, that deleted mitochondrial DNA is actually expanded. And it clonally expands in those cells. So in a muscle, which is a syncytium with many different nuclei, you get bands of cytochrome oxidase positive, negative, as if there was a clonal expansion of those mutants. Interestingly enough, in the bone marrow, where if you are heteroplasmic for deletion, in fact, those cells with less deletion have a replicative advantage, and the blood cells become negative for the deletion. So there's no deletion in blood, even though it goes to 70 or 80% in muscle and brain. So you get this divergence of genotypes by these replicative advantages. We also see both positive and negative replication for point mutations, but we don't understand how that occurs. And I'm going to tell you about some really weird things tomorrow.
Why don't we take one more?
Great. Thank you for that great talk. So I would like to hear your thoughts about cytoplasmic and more specifically mitochondrial donation from younger reproductive age women to older reproductive age women in vitro fertilization.
Thank you. You may be either pleased or distressed to know that last week there was an Institute of Medicine hearing on that subject, of which I was asked to testify, and every time I say something, I get in trouble. So I can assure you that I took a lot of grief. Where to go? There are several attempts at, okay, so 2 issues here. One, if you have a maternally inherited disease, a woman with a mitochondrial mutation, you can be like the first lady that I told you, where all of her 3 children with 2 different husbands are going to die. Okay, so that's devastating. So one idea that actually I proposed in the 80s, so it's partly my fault, is the logical thing to do would be to take the nucleus from her fertilized egg and just stick it into an ovum which had good mitochondrial DNA. So then you would simply eliminate the problem. Well, it turns out that that's now a technical reality that can be done either by the nuclear transplantation or by a man named Mitochondrial in Oregon, where he can actually take the oocyte spindle and move it over to the to a new oocyte and fertilize it. And he's gotten completely viable, apparently normal macaque monkeys. Okay, so for women who have high levels of mutant mitochondrial DNA such that every child will die, the only hope for those women to have successful reproduction with their own nuclear genome will be this nuclear transplantation. I was asked to talk about mitochondrial lineages, and everybody hoped that I would say that they're all the same and there's no genetic difference between mitochondrial lineages, and I had to tell them I didn't believe that. So then the question is, well, is that going to be a kind of gene therapy? And I suggested, well, why don't we just match haplogroups, so then that problem goes away. Well, we'll see where that goes. But the real issue from an in vitro fertilization point of view is advanced maternal age. That's really what's a big problem in vitro fertilization. And one hypothesis is that the reason that older women have trouble conceiving is that they have old egg cytoplasm. Maybe old egg mitochondria, although you don't necessarily have to argue that. So if this technology could be used for curing maternal inherited disease, why not just use it for all the older women? Okay? And there are a number of corporations that are pushing hard for the development of this technology because they see big dollars in doing it. Okay? I argued for a moratorium on that until we found out just how important mitochondrial haplogroups are and whether it will make a difference, because we don't know. But I do think that the cost benefit for women with severe mitochondrial disease is justified to try it for them. But I don't think we should try it for tens of thousands of women. And so I'm sort of in the middle here on that. Of course, there are some people that think we shouldn't do it at all, and other people think we should do it for everybody. So that's where we are on that.
So Dr. Wallace will be around for about 10 minutes till we have to switch him off to dinner.